so grateful to the Grays and the Potters for being so gracious. Your vision carries a lot of weight. It's really inspiring. Amid all the uncertainties, Joan just pointed out about funding, about future of our trainees and so on. This is really a bright ray of light, okay? I hope I'll be able to deliver in such a fashion so that you learn a few things about what we have been doing in the past 30 years or so, in fact. Okay, time is of essence, so Susan, would you mind giving me a little sort of like a, sign if I go on too long?
I will.
So like a few minutes? Okay, thank you. I know you will. I know you will in a very forceful way too. Okay.
I know what that sign looks like.
Okay, all right. I am not the right person to talk about cancer genetics. I wasn't trained as a cancer geneticist, although I would have loved to be a cancer geneticist, but it's too late for me to go back to school to learn cancer genetics. But when I get low, when I get depressed or get a little bit discouraged because my grants are rejected by funding agency, papers are rejected, or reviewers suggest 5,000 experiments for you to do, I pull this up as the source of inspiration and motivation. Look at those dreadful numbers. Our colleagues in the audience can tell you more precisely what these numbers mean, but to me, we've got a long way to go, so I'm going to keep going. Hopefully we can make meaningful contributions to help alleviate the burden, the cancer burden, imposed by BRCA mutations. It's more than just BRCA mutations. There are a lot of genes that function together with BRCA. When mutated also would lead to breast, ovarian, and other types of cancer as well. What do we know about BRCA1 and BRCA2? It's a very long story. I'm going to filter it. I'm going to boil it down for you, okay? Long and short of this, there are not shortage of publications on this. There are 45,000 publications on BRCA1 and BRCA2 and related genes. You can legitimately question, then Patrick, why are you doing this if you have 45,000 publications on it? And so I try to convince you today there's still a lot that we don't know about BRCA1 and BRCA2. From the standpoint of understanding the involvement in DNA damage repair, repair and preservation of damaged DNA replication forks when cells undergo cell division, you need to replicate your DNA. A lot of problems arise then, without going into great detail because of the nature of the audience here. BRCA1 and BRCA2 also involve in the resolution of harmful nucleic acid intermediates called R-loops, I should say. We're working on this, but I don't have time to talk about that. Actually, we have a paper in revision in a very good journal. It's taken 14 months already, so we're about to resubmit concerning the role of BRCA1 in R-loop resolution. Because of the role in R-loop resolution, they help avoid transcription replication conflicts. There's quite a bit of evidence that BRCA1, possibly BRCA2, involved in transcription as well. We don't work on that because there are people who can do this 100 times better than we could. We're supportive of them, but we don't actually look at the transcriptional aspect of these things. So this is what I'm going to structure my presentation today. I would try to tell stories rather than delivering a very technical presentation that would boggles your mind and boggles mine too. Okay, I'll try to be as simple, as understandable as possible. Marie Jason and also Maggie gave an incredible introduction about what BRCA-dependent homologous recombination is. So I'm just going to briefly touch upon that instead of duplicating what they've done. I'm going to take a few moments to tell you what BRCA2 does, as far as we know what it does, in terms of helping a protein called RAF51. I'll give you some background what RAF51 is. Why is it a big deal to study it as I go along? And then I will switch gear a little bit and talk about the role of BRCA1 together with this partner protein called BART1. So when I talk about BRCA1, it's really a dimer, okay? BRCA1 partner with BART1 to perform certain biological functions. How they influence RAF51 activity, how they actually affect the processing of a DNA double-stranded break to generate the template that's used by the DNA repair machinery to perform the repair reaction. And then finally, I'm going to talk about some studies or part of the studies that we've done very recently. Some of it are currently in present science about how BRCA1 BART1 together with the complex of proteins classified under the 53BP1 axis, how they antagonize one another to determine whether the lesion should go into homologous recombination for repair or go into a different pathway for repair. I try not to be too technical as I go along. Unfortunately, even though I'm trying to be very, simplified here, okay, for instance, the 53BP1 complex, I have to learn how to use this point that's massive, okay? I'm grossly simplifying, okay? 53BP1 complex consists of maybe a dozen or so proteins. What they do is they occlude the DNA ends. Why do they want to do that? The goal is to prevent homologous recombination from working so that this break can be channeled in a different DNA repair pathway called DNA end joining. I'm going to come back to comment on that later on. And as you can see, it's very complicated here. These are nucleases that are involved in processing the DNA ends to generate a piece of single-stranded DNA for loading of the RAF51 recombinase protein, as I've shown here. BRCA1 BART1 has a major role to play in this stage of the DNA repair reaction. Likewise, BRCA1 BART1 together with BRCA2 also have a very important role to play to load RAF51 onto these so-called resected DNA ends. So that RAF51 can go and look for a homologous piece of DNA to do the strand invasion reaction. This particular structure is called displacement loop. So when you hear displacement loop, this is the intermediate you need to conjure up, in your head. So the reason why RAF51 makes a D-loop like this, or displacement loop, is to create a primer template junction to prime DNA synthesis. I'm not going to get too technical. Once DNA synthesis has occurred, it can be resolved by different recombination pathways with very different outcomes. I'm not here to talk about DNA repair pathway choice, so I'm going to stop there, to focus on BRCA biology. What are the tools at our disposal? We use all the organisms. We don't do fly, We're not a fly lab, but we do yeast work, Up until 10 years ago, we were a yeast lab, and then we made a fateful decision. So we switched to human and studied BRCA1 and BRCA2. That was sort of foolhardy because we didn't have any grant funding on it. We have to divert a lot of resources in order to support this. We made a decision, never looked back. Great decision to make, Wasn't a mistake for sure. We're considered leaders in studying nucleic acid biochemistry and enzymology. We are one of the notable groups to be able to reconstitute very complicated biochemical reactions. Sometimes we have 15 different proteins, in the test tube. We also known for our ability to look at DNA transaction reactions at a single molecule level using single molecule biophysics and also structural biology. We are by no means great cell biologists, but we're competent enough to be able to do some cell biology and some simple genetics as well. All right. Did I catch you by surprise, mating type switching? I will ask you a question. How many of you in the audience know what mating type switching in yeast is? 1, 2, 3. Wow, not too many. Roger, do you know how it works? You know what it is, but do you know how it works?
HL.
Yeah, there you go. Thank you. He knows. I wasn't pulling his leg. So I was teaching this stuff at Yale for 16 years. All right, so pretend you're my undergrads. All right, we're not charging you tuition. That's a comfort, right? Be very brief. So we're talking about the biggest yeast used to make bread and so on, Saccharomyces cerevisiae. Quite a mouthful. They can exist as haploid. That means that they have only one chromosome, of either A or alpha mating type. 2 haploid yeast cells can mate, but only cells of opposite mating type can mate. You cannot make an AA diploid or A alpha diploid or alpha alpha diploid, but you can make an A alpha diploid, so to speak. Why am I telling you about this? This is actually initiated by a DNA recombination event, Initiated by site-specific DNA that was rendered by the HO endonuclease. Roger got it right. All right. So the mating type switch reaction is successful execution requires genes of the RAF52 epistasis group that are involved in DNA double-strand break repair. That's the relevance. That's why yeast genetics is actually very, very important. So how does it work? Without going into detail, if you're really into great details and so on like I am, If you cell of a A mating type, the A mating type, you will go to a locus on chromosome 3 called HML alpha to look for genetic information, okay, to convert the red information to blue, okay? If you of MET alpha mating type, you will go to HMR, okay, for the donor information and so on. The whole process is initiated by a site-specific DNA double-stranded break introduced by an endonuclease, which is an enzyme that cuts DNA on chromosome 3. A lot of genetic analysis have shown that proteins and genes of the RAF82 epistasis group are not only important for mating type switching, but they present incredible model for us to study what happened to the break, right? Because you can watch it by cell biology, you can do chromatin immunoprecipitation and all types of tricks with a simple organism like yeast. A lot of genetic analysis have shown that members of the RAF82 epistasis group not only require mating type switching, but absolutely needed for DNA double-strand break repair, homologous recombination, and meiosis when you make your germ cells. We're not going to talk about meiosis today. That's an entirely different ballgame, maybe some other time. Okay, prominent members of the RAF82 epistasis group are listed here. We've published extensively on all of them over the past 20-some years or so. Won't have time, nor is the purpose to go into all of that. But I'm going to pick two guys out, okay, to focus on. RAF81 and RAF82. The purpose of that will become very obvious, okay, in just 1 minute. All right. This was a year and 4 months after I became an assistant professor at the University of Texas Medical Branch in Galveston. All right. If you take Interstate 45, you fly into Houston, keep going until you can't go anymore. Okay, because you'll hit the Gulf of Mexico if you keep going. That's Galveston. Okay, wonderful medical school. Well, wow. I get banned because of that, right? I'm only kidding. Okay, all right. But this is like a year and 4 or 5 months after I became an assistant professor. I was able to demonstrate that yeast RAF81 protein has the activity expected of a general recombinase, being able to take DNA, shuffle it around, okay, and so on to do the DNA repair. Single author paper in Science. That was the only person working in my lab myself, okay, at that point. I didn't have a graduate student, technician, what have you, and so on. At that time, it didn't really quite strike me what the significance of this is. But I thought, well, somehow it would be good for my career development, maybe for my tenure, maybe get my grants funded. Both of them proved to be true, but I didn't know the system that we developed one day would become important for testing BRCA1, BRCA2, and related proteins, as I will show you as I go along. So that's the history. Just briefly, how does it work? If you incubate RAF81 with a piece of single-stranded DNA, it's circular single-stranded DNA. For the people who know molecular biology, it is an M13 molecule, about 7 kilobases in length. RAF81 will form this really incredibly highly structured filament on the single-stranded DNA. You can do three-dimensional reconstruction of these filaments. Okay, if you really the type who would like to know all the biophysical attributes of this, it's a helical filament of a right-handed helicity with about 6 monomers of RAF81 tying about 18 bases of DNA. The important thing to remember is that within this structure, the single-stranded DNA is stretched, okay? We describe the stretching by the increase of so-called axial rise, which describe the distance that separates 2 bases in the DNA. In B-form DNA, the axial rise is 3.4 angstrom. If you're into that type of stuff, it becomes 5-point-something angstrom, stretch 50%. The reason why it wants to do that is to expose all the DNA bases. When you do DNA recombination, you can pair, all right? Just put them very simply, all right? So am I making this up? I'm not, okay? We've done a cryo-EM reconstruction of the intermediates where the single-stranded DNA bound by RAF81 go and look for a homologous DNA sequence, and we were able to capture the intermediate. That was so exciting. Okay, the downside of this is that this structure is not of high resolution enough for us to tell all the atomic details and so on. Structural biology is all about resolution. All right. 3 angstrom makes a huge difference than compared to 6 angstrom resolution. This structure is about 9 angstrom. We have some distance to go. All right, so that's RAF81, so that's good. But RAF51 binds DNA very slowly. The so-called rate-limiting step of the whole reaction. It takes all day for it to get on DNA. It doesn't take all day. It takes many minutes, okay, to put it that way. Because it takes so long, okay, it's prone to interference by a single-strand DNA binding protein called RPA, or replication protein A. RPA is a short form of it. It's very abundant RPA, about 10,000 molecules per nucleus, extremely abundant, ubiquitous, from yeast all the way to humans, highly conserved. So it occupies the single-stranded DNA, and that's bad, because you need to roll RAF51 on the single-stranded DNA for it to work in the DNA repair reaction. So a group of mediator proteins, very generic here. I'm not saying this is this protein or that protein, but generically, there are a group of proteins called HR mediators, homologous recombination mediators. They take RAF51, physically load it onto DNA to allow it to bump RPA off, to assemble this RAF51 filament structure that I've shown you some EM micrograph and reconstruction just a couple minutes ago. All right, so the HR mediators in Saccharomyces cerevisiae, more than one. The major one is the protein encoded by the RAF52 gene. We were first to report this, okay, in 1997. Just a couple of months later, there were a couple of papers in Nature by our competitors at the time, we're friendly, okay, published in Nature about the same phenomenon, so to speak. So RAF52 is a crucial HR mediator. In addition, there's a series of RAF51-like proteins. They don't do what RAF51 does, although they're structurally related, called RAF51 paralogs, for instance, RAF55, RAF57. They play very important accessory role. Because of time, we're not going to go into great detail. As a result of the role of RAF52 as the major mediator, if you mutate it, if you get rid of it, E cells become extremely sensitized to different types of DNA damaging agents that directly or indirectly cause DNA double-stranded break. So including mating type switching defect, by the way, okay, emphasizing the utility of studying mating type switching. Now, RAF52 interact with RAF51 physically and also with RPA physically and bind single-stranded DNA. So that's a chaperone, so to speak. It takes RAF51, bind DNA, and bump off RPA, so to speak. Okay, so that's yeast. Now, there's a yeast ortholog, or yeast equivalent of RAF52 in mammalian cells, including human cells. But the thing is, you can delete it. Cells seem to be quite happy, okay, even without it, telling us that it's got to be some other protein molecules, okay, doing the RPA RAF51 exchange. You've guessed it already. Which one is it? It's BRCA2. I'm going to tell you a little bit about exactly how BRCA2 does it. Well, not exactly, but what we think mechanistically it is doing. Okay, point out some real difficulty here. BRCA2 is incredibly large, about 400 kilodalton, over 3,400 amino acid. For people who don't think about this, okay, this is like an elephant compared to your cat. Okay, this guy is huge. There's a lot of very interesting features about, I'm going to tell you about some of it. Now, BRCA2 physically interact with RAF51 and also bind single-stranded DNA, but it doesn't interact with RPA. That sets it apart from yeast RAF52. We have found that a very small polypeptide that's highly acidic is only 70 residues in size, but 27 out of 70 residues are acidic. It's acting as a DNA mimic in that particular regard. All right. Now, in addition to BRCA2 together with DSS1, there's considerable evidence suggesting that BRCA1, BART1, PALB2, and a number of RAF51 paralog proteins function together with BRCA2. This is a major challenge moving forward to figure out, what the big complex is doing. We're making very good progress. Okay, if I see you at a conference and so on, maybe I have something to report them. I just want to point out, I try to cite work done by others as much as I can, in addition to our own work. I apologize if I fail to cite one or two key papers, okay, in this particular regard. BRCA2 is a very large protein molecule with two known DNA binding domains. We found this new DNA binding domain reported in Nature Communication a couple of years ago. There's a low affinity DNA binding site within the N-terminal region of BRCA2. We don't know exactly what it does, okay, in terms of contributing to the mediator activity. Has all these so-called BRC repeats called breast cancer repeats that interact with RAF51. There's a RAFD1 interaction domain at the C-terminal region encoded by exon 27. Rob was talking about exon 27 cell model. We're very excited. I think we can work together on this, and so on. We have published extensively, as have others, okay, showing what BRCA2 does. Because of time, okay, I'm not going to go into actual technical detail. Just to sum it up, okay, RPA occupies DNA because of its high affinity for single-stranded DNA. BRCA2 together with DSS1. DSS1 acts as a DNA mimic. BRCA2 actually engages a single-strand DNA substrate, and altogether they synergize to replace RPA by RAFD1, so that recombination DNA double-strand break repair can occur in a timely fashion. Right? We published a paper in Molecular Cell. Please go read it if you haven't read it. Yeah, thank you. We try very hard to build up all these bigger complexes to try to figure out what they do within the context of loading RAFD1 on DNA. It's a huge problem to load RAFD1. That's why cells have devoted a lot of resources, okay, in order to do it. In this particular regard, collaborating with Kara Bernstein, who is a faculty here, senior faculty here at Penn, and also with Marie Jason at Sloan Kettering, Memorial Sloan Kettering Cancer Research Institute, we have solved the cryo-EM structure of two of these RAFD1 parallel complexes. One was published in Nature a couple of years ago. The other one we're putting together right now. We don't know where to send it, okay, but it will be a good journal. Okay, hopefully we'll be able to report, okay, some good news shortly. Okay, so that's about BRCA2. You can ask me about any BRCA2 question later on, so on. Now we want to go back and talk about BRCA1 together with its partner, BART1. Okay, so that's a linear, okay, representation of BRCA1. It's fairly large. It's not as large as BRCA2. I would say this would be like a giraffe, not an elephant maybe in terms of size, okay, but quite large. BART1 is smaller, a little under half the size. Now it has these very well-recognized and well-folded motifs and so on, including ring domain. They form this dimer, or they form this partner complex through the N-terminal ring domain. It's 1-to-1. That's the stoichiometry, all right? The BRCD domain is involved in protein-protein reaction. The coil is involved in binding a protein called PALB2. Some people even call it a BRCA3, and so on, all right? So all of that is great. There's substantial amount of structural information on the BRCD domain and ring domain because they're well folded, okay? They look great, useful information. But the most interesting part, as far as we're concerned, is actually on structure. In other words, it doesn't have a real rigid structure flopped around in solution. It's impossible to study, okay, structurally, but we can get around by NMR. This took a long time to solve. So we do all the necessary labeling and so on, did all the MR analysis. I'm showing you just one example of being able to map out the DNA binding domain. This is really interesting. Okay, hear me out. If we put in double-stranded DNA, we have group of residues that light up. More than likely, they're involved in binding double-stranded DNA, but they don't seem to be involved in binding single-stranded DNA. But we have another group of residues that appear to be specifically involved in binding single-stranded DNA. Okay, that's all I'm going to have to say about this, but this guides our ability to make specific mutants and explain why certain cancer mutations affect DNA binding and protein activity and so on. All right, so what do we know about BRCA1 BART1? Not complete, okay, but we know several things about it. It's a so-called E3 ubiquitin ligase activity. Take a protein molecule, ubiquitin, you conjugate it to intracellular targets. We don't know what the substrates are. We're working very hard to try to find them. BRCA1 BART1 also binds DNA, directly upregulates the activity of RAFD1. As I alluded to before, BRCA1 BART1 forms a higher order complex with BRCA2 and with PALB2, which is another breast and ovarian tumor susceptibility gene. We have been able to reconstitute that very big complex. Initial results are very, very encouraging. Let me go back one. Okay, don't have time to talk too much about that. BRCA1 BART1, based on our recent work, is also involved in regulating the initial DNA end resection reaction to generate a single-stranded DNA template for the assembly of the RAFD1 filament. We call this a licensing role because once you resect the DNA break, more than likely it cannot go back to non-homologous DNA end joining for repair. It's involved in allo resolution, don't have time to talk about that. It affects transcription. I already told you we don't work on this because there are people who know a lot more and can do it a lot better than we can. Roger Greenberg, studying in the audience, he and others have found several other complexes containing BRCA1 with other nuclear factors. They do completely different things, all right? It's not my area of expertise. You can ask Roger about that. So in 2017, a very highly motivated, who's a tenure faculty now, developed a procedure for purifying BRCA1 BART1 as a heterodimer, 1-to-1 complex, and carry out a lot of biochemical analysis. I cannot show you everything. Okay, so it binds DNA, especially the so-called displacement loop structure. There's a structure that's made by RAF51, followed by DNA replication fork-like structure, double-stranded DNA and single-stranded DNA. All right, what does it do to RAF51? So if you set up a so-called D-loop reaction, this is just to mimic what's going on in cell. You will assemble RAF51 onto single-stranded DNA. It's just a 90 nucleotide, okay, single-stranded DNA oligo here, and throw in a large plasmid molecule with a single homologous site. So RAF51, together with BRCA1 BART1, can go and look for the homologous site to make the D-loop structure. The D-loop is run in a very different spot in the gel, and you can quantify and so on. Immediately, you see that BRCA1 BART1 greatly upregulates the ability of RAF51 to make this D-loop structure, about 20 to 30-fold. This is very highly biologically relevant. All right, okay. So this is all I'm going to have to show you about RAF51 BRCA1 interaction. If you're interested, go and read the paper. It was published in Nature in 2017. Just to sum up some of the things, others I don't have time to show you. The DNA binding activity of BRCA1 BART1 is indispensable in this regard, as you might have expected. Both BRCA1 BART1 physically interact with RAF51, and this attribute of the tumor suppressor complex is likewise indispensable for RAF51 enhancement. Now, this is not our work. I wish it was, but it wasn't. Someone found that, Joe Morris, okay, in the UK, found that the BART1 RAF51 complex that we first documented in our Nature paper is also very important for preserving replication forks that have been damaged. It's not our work, it's Joe Morris's work. Beautiful work. In the last part of my presentation, I would want to take you back all to the beginning, when the DNA lesion must be processed in a unidirectional manner to generate a single-stranded DNA. I need to take my time to explain because this is unnecessarily complicated. Biology fools us sometimes. It's why is it so complicated? We can figure out. We could not. It's bad because it's so complicated. It's good because that means that I have a job for a while, trying to figure it out. All right, so again, pointing out that double-stranded break are occluded by 53BP1. It's a big complex of protein. I'm going to come back to comment on that. If 53BP1 group of proteins successful, this occluded end is destined for repair by end joining, abbreviated as NHGJ, non-homologous DNA end joining. But with the help of BRCA1 BART1, somehow this complex is evicted, okay, from the DNA end. So now the ends are accessible to nucleases that process the end in a very unidirectional fashion. Only the strand that contains a 5' end is chewed away. Now, this is really complicated. There are 4 machineries, okay, that would perform this end resection reaction. One consists of 4 different proteins called RAF50, MR11, MBS1, together with a protein called CTIP. Because of time, I won't go into great detail. And then 3 other machineries, including a 5' to 3' exonuclease, XO1, and also a DNA helicase, either BOM or the helicase Werner, together with DNA2, a structure-specific DNA endonuclease, will actually catalyze long-range resection reaction, generating DNA that is maybe several kilobases in length, all right? Incredibly complicated. Because of time, I won't go into all 4, resection machinery. Suffice it to emphasize that if resection is successful, that resected end is destined, okay, for BRCA-dependent homologous recombination. All right, so what does BRCA1 BART1 do in DNA end resection? About 4 or 5 years ago, if you talk to people in the field, they would tell you more than likely somehow it's involved in antagonizing the role of 53BP1 in terms of occluding the DNA end, but probably doesn't really directly get involved in the resection reaction. Okay, there's only one way to find out for sure. It's to do biochemical reconstitution. I hope this is compelling to you. So we purify all the protein factors needed for the reconstitution. A research track faculty in my lab spearheaded the effort. It is brilliant. So you can see immediately, titrating a small amount of BRCA1 BAT1 greatly upregulates the ability of BRM to unwind the DNA substrate. This is important because you generate, okay, the DNA intermediate, important for generating the single-stranded DNA. So just imagine DNA is being separated, the two strands, DNA2 would nibble, nip, nip, nip. Ultimately, you have a single-stranded DNA, there are 1,000 bases in length. So this is DNA unwinding. What about the actual DNA and resection reaction? Okay, so this is the rate without BRCA1 BAT1. That's the rate together with BRCA1 BAT1. So there's up to 10-fold upregulation of DNA and resection by BRCA1 BAT1. So BRCA1 BAT1 is directly involved in the end resection reaction. Consistent with that, we see protein-protein interaction, okay, by standard pull-down using purifier proteins. And BRCA1 BAT1 actually doesn't affect DNA2 activity in this particular regard. All right. Here, we use a completely different methodology to look at upregulation by BRCA1 BAT1 using the so-called DNA curtain system by our collaborator Eric Green at Columbia University. He's a brilliant biophysicist. He worked out the chemistry to load a cover slide with maybe 100 different molecules, okay, of a DNA. By strategically labeling the DNA or labeling the protein factors, you can track the movement, okay, such as a Bloom helicase on DNA. This is called chymograph. Basically, it's distance travel over time. You can decorate the resected DNA with M-cherry tag RPA, which appears to be red. Okay, so you can do multiple different things to look at movement, okay, to look at resection and so on. Then look at enough of them. You can calculate the rate with, the proper statistical, okay, rigor and so on. Now, without BRCA1 BAT1, okay, that's, okay, the velocity, how fast, okay, the machinery is moving. Titrating in BRCA1 BAT1, as you can see, the rate increases significantly. Not only the rate increases significantly, it travels longer distance. So you turn on the turbo, not only the car goes faster, it lasts longer, okay, so to speak. So there are two different things going on here. I don't have time to show you BRCA1 BAT1 upregulates Werner as well, the other DNA helicase, and also AXO1. Don't have time to show you all the data. Please go and read our paper, humbly. Okay, all right. Before I close up, I want to address another very important issue that has major implications about drug resistance. We all know that PARP inhibitors, under some circumstances, work quite well, okay? But the downside is that many patients who are initially responsive will develop resistance, okay, over time. What are the resistant mechanisms? We don't know for sure, but this could be one of the explanations here. It goes like this, all right? So I already talked about the resection factor, the role of BRCA1 BAT1. Let's go and look at, okay, proteins of the 51 BP1 axis, including RIF1. Shielding is a tetramer. I'm not going to go into the detail. There's a trimer, trimeric complex called CST, STM1, CTC1, TAM1. Tisha Delan is the guru in terms of studying this particular complex, what it does at the telomere. It does wonderful, beautiful, intriguing things at the telomere. You can ask her for that. And actually, Tisha was the first group to propose that through the recruitment of alpha primase, this DNA polymerase alpha, together with primase, it fills in receptor DNA ends to slow down the resection reaction and perhaps in some other way to restrict, okay, HR. Great idea. Very consistent with what it does at the telomere. And we're rigorously pursuing this particular idea. And our initial findings are very consistent, okay, with some parts of that particular model. But we want to ask a different question. Do CST complex and other circulating BP1 axis factors actually directly regulate DNA and resection? independently of recruiting pro-alpha primase to resect the DNA. And it's a simple question. We have the tools to answer that question. I wouldn't be standing here talking if we didn't find anything. We found something, okay? I want to walk you through. This one is not published yet, but should be appearing in Science in maybe 2 weeks or so, okay? Cody Rogers is a very talented postdoc. First one in his family to go to college. Is from the Midwest. I'm trying to convince him to stay in academic science because he will have a great future. All right, okay, I won't be...
No, bring him to, we'll take him at Penn.
Okay, okay, all right, good. I will let him know, by the way. Okay, so he developed procedures for purifying CST and multiple DNA binding mutants, okay? So the DNA binding surface is huge. So it's been reported that OBF and G are involved in DNA binding. We know that at least OBD is also involved. We have made mutants and so on. But for this purpose, okay, I'm going to describe 2 mutants to you. One's called M5, the other one's called M10. Essentially, just 5-point mutation, M5. M10 would be 10 different point mutations based on published work by Domchek that was published in Science. As you can see, these proteins can be all purified to near homogeneity. By a mass photometry, we can show that they are monodispersed. Important to show that they're monodispersed. They don't form soluble aggregates and so on. By MST, which is a quantitative way to look at DNA binding affinity, both mutants show an overt, okay, impairment. Not a defect, okay, because there are other OB4s involved in DNA binding, but an overt impairment in DNA binding. If you're interested in the kinetic constants and so on, you can look down here. All right, so with these tools, we ask the first question. Does CST directly restrict exon-1 activity? That's a 5 to 3' exon. So we use a 2 kilobase pair double-stranded DNA, randomly labeled in the middle with P32. Okay, when the protein digests the DNA, it generates this intermediate and finally a very small product, which can quantify by measuring the amount of DNA that remains, that sort of thing. With wild-type CST, a small amount of CST is sufficient to shut down the end resection reaction. Compare lane 3 to lane 6, from having about 90% resection to almost nothing. Okay, this is wild type. The M5 mutant shows a partial defect in this particular regard. Look at lane 9. Okay, the M10 mutant, which is more defective in DNA binding, correspondingly shows a stronger defect in this particular regard. So DNA binding is important. DNA binding by CST is important for restricting exon 1. That could be the end of it, but no, it gets more interesting. All right, then we look at to see if exon 1, okay, blockade can be reversed by the addition of BRCA1 because genetics and cell biology tell us that BRCA1 somehow does something, okay, to overcome inhibitions by CST. Okay, lo and behold, titrating wild type with BRCA1 is able to completely overcome the inhibition. Compare lane 4 inhibited, lane 5 restore. Okay, very efficient in restoring. I have to back up, all right? And then we tested 3 DNA binding mutants of BRCA1 complex. Long and short of it is that we can conclude that DNA binding by BART1 is indispensable. The DNA binding activity of BRCA1 is important, but not as indispensable as that of BART1. The double mutant has more of a defect in lane 14, as you might expect. And there's protein-protein interaction also. We don't have time to go into great detail. I'm showing you the data so that you know we actually did the work, okay, in this regard. Okay, that's exon 1. What about BRMDNA2? Some surprises here again. First, CST strongly restricts DNA resection by BRMDNA2. You'll see in a minute that's the case. But very surprisingly, to do DNA binding mutant of CST, okay, are completely proficient. They have no trouble inhibiting BRMDNA2, quite unlike the case of exon 1. So some other attributes of CST must be important for restricting BRMDNA2. All right, and don't have time to show you all the data. CST does not affect DNA unwinding by BRM and has a modest effect on the endonuclease activity of DNA2. And also, addition of BRCA1, even in excess of it, cannot alleviate, okay, the block imposed by CST. Then we came up with the hypothesis that perhaps protein-protein interaction between CST and BRM somehow could sequester, okay, the BRMDNA2 complex in an inactive way. You can easily imagine DNA2 normally is positioned DNA is unwound, it can nip, unwind, nip. But if you twist it out of the way, even though you have unwinding, there will be no nipping anymore. So based on that particular premise, we look by AlphaFold prediction and so on and so forth. We predicted a basic pocket in the OBB fold of CTC1 could be involved, interacting with acidic patch in BLM. So Cody, together with our structural biologist colleague, Sean Olsen, went ahead and made 2 mutants. They made only 2 mutants. Both of them worked. When was the last time you made 2 mutants, both of them worked? Okay, so this is amazing. I don't know how they did it. Actually, I don't care how they did it. They did it, okay? I'm very happy that they did it. They explained to me, but I go, oh, okay, good. It works, great. I'm very excited about this. So this KRR mutant and KQK mutant basically designate 3 amino acid, being changed from KRR to alanine or KQK to alanine. Important, to show that they bind DNA just fine, no problem. They bind DNA just fine, but they greatly impair for the ability to interact with BLM. So this is a good mutant, a good separation of function mutant to test in our reconstituted system. Oh, this is so gratifying. Okay, so if you look at the CTC1, KRR, the KQK mutant, compared to the wild type, you see that the wild type doesn't inhibit DNA unwinding very much. It's indicated by accumulation of single-stranded DNA in this section reaction. But the wild type CST strongly restrict, the ability of this BLM DNA2 ensemble to resect the DNA, as represented by the red curve here. Both the KRR and the KQK mutant are overtly defective in this particular regard. So protein-protein interaction involving OBB, rather than the DNA binding attribute of CST, is actually important, for restriction of BLM DNA2. Now, this is all biochemistry, which is fine and good, right? But any reviewer, who's serious about this will ask us, show us some biology, Show us that in vivo, your mutants behave in the fashion that you think they behave. So we pair up with Dipanjan Chowdhury, our collaborator at Dana-Farber Cancer Institute. He has a really outstanding postdoctoral fellow, Michelle Swift. I can only show you about 10% of the data generated by Michelle because of time. Michelle did a bunch of very critical experiments looking at the 4 mutants of CTC1, the M5 mutant, M10 mutant, KRR, KQK, All of them, show a hyperresection phenotype, meaning that the ability of CST to restrict DNA resection is gone or impaired in these mutants. But more importantly, if you look closely at the data, I will point you out, where you should be looking. The M5 mutant, remember, showed the least defect in terms of restricting XL1. It showed the weakest phenotype. There is a phenotype for sure, but weaker. The M10 mutant is incrementally more, impaired and show an even stronger phenotype. And the KRR and the KQK mutants are the most impaired in vitro. Accordingly, they're also the most impaired, within the cellular setting. I'm almost done, Susan. All right, good. Now, one very famous phenomenon or important phenomenon is that if you have a BRCA1 tumor, it's sensitive to PARP inhibitors and so on, but if somehow you can restore enough homologous recombination activity, for instance, by mutating 53pp1 of any of the proteins associated with 53pp1, the residual activity that's restored, is able to confer resistance to PARP inhibitor. So we look at PARP inhibitor sensitivity. Again, the 4 mutant backgrounds and so on. Controls down here, knockdown, complemented, with the vector and so on. The M5 and N10 mutant, render cells PARP inhibitor resistant, but not as much as the KRR and the KQR mutant. Again, you have a greater response, corresponding to the ability of these mutations to impair the ability of CTC1 as an inhibitor of DNA resection. It's still correlative, but I think it's pretty strong correlative evidence what's going on. Let me give you a synopsis. I can only cherry pick a few things to tell you. CST not only recruits pro-alpha primates to DNA breaks, it's very interesting here. We're actively pursuing what the end filling, okay, is doing. We're finding things that are quite interesting in this particular regard. But what I've shown you in the past 10 minutes or so indicates that CST also acts directly to restrict DNA resection per se, independently of pro-alpha primates in this regard. A number of biological consequences, okay, result from this lack of resection inhibition. You have hyperresection of DNA breaks. You have attenuation of non-homologous end joining as predicted, HR, homologous recombination enhancement, and alleviation of PARP inhibitor sensitivity in a BRCA1 deficient background. All right. And also, I need to point out that BRCA1 part 1 can very efficiently overcome the AXL1 blockade, but it must act via another mechanism to restrict, okay, a BRMDNA2, or to overcome the restriction on BRMDNA2. I misspoke a little. Okay, what is it? Is that because of ubiquitin conjugation or some other mechanism? We don't know, but we're testing all possibilities. To everybody in the audience, okay, as you know, science sometimes is by process of elimination. You have 10 hypotheses, you do 10 experiments, you eliminate 9 of them, boom, you come to the truth. Okay, rarely you come up with a flash of brilliance, okay? The first thing that comes to your mind turns out to be correct. But it doesn't happen very often, I must say that. So this paper is in Press in Science. Actually, we had a very easy time. Reviewers were quite enthusiastic about this. Suggested maybe 3 months worth of experiment instead of like a year, 15 months, that sort of thing. I think my colleagues know exactly what I'm talking about here. So where do we go from here? This is my last slide before I do some acknowledgements and so on. All right? We are well poised, I believe, to continue making important, or I shouldn't say important, you judge, significant contributions in elucidating how BRCA1, BRCA2, and associated protein actually work in DNA damage repair, in out-loop resolution clearance, avoidance of transcription replication conflicts, and replication fork preservation in general. We have ideas, okay, we're carrying out experiments along this line. I think we're well poised to continue making some contribution in this regard. Together with colleagues, okay, at UT Health San Antonio, we're looking at other etiological factors, including obesity, which is particularly prevalent in South Texas, and genetic modifiers. There may be a gene downregulated, upregulated elsewhere that regulate, okay, cancer susceptibility, okay, in BRCA carrier. Specific to South Texas, we have a mission, we have goal, okay, to do things that will benefit South Texas, and in general, everybody too, hopefully. We will pursue drug discovery and development rigorously. My dean is sitting in the audience somewhere. Oh, he's such a pain. Said, Patrick, do drug discovery. Patrick, you should do more drug discovery and so on, and we're doing it, okay? It's worthwhile to do it, okay, but he's a real, okay, expert in this particular regard. I'm kidding, of course. You know, we'd love to do it with him. He's a great guy. Okay, so this is my current group. Okay, just want to point out people whose work I've touched upon. Cody Rogers, he comes on the job market pretty soon. The guy with the red arrow right here. Angie Jasper, don't have time to go too much in what she's doing. She's the student about to graduate from the MSTP program. Essentially, they get 2 degrees, MD and PhD. She's brilliant. Okay, it's sad to let her go. But all good things have to end, okay, so to speak. And we have 3 research faculty, Young Ho Kwan, Samir, and also Yush Pao. They're brilliant, okay. They're not low maintenance. They are no maintenance. You don't ever have to do anything, okay, to get everything. Because actually, if I show up, I screw things up. I try to stay away as much as I can in this particular regard. These are my colleagues, former trainees, and as you can see, we have been able to build incredibly synergistic, okay, situation with all these investigators are funded by the National Cancer Institute. Really quite precious, in my view. We support each other, you know, very much. And together with Chowdhury Lab and also the Green Lab at Columbia, we have a newly funded, okay, a program project grant from the National Cancer Institute that was 6 months ago. If it's now, I don't know what will happen. We got it funded 6 months ago and hope that will continue. With that, I will stop.